Welcome to Insight PurePRP. In this short video, you will learn the basics of preparing PurePRP with the world's leading concentrating system. Before we get started, you must remove the red vented caps from the self-sealing port. Then, swab the port with sterile alcohol. Always swab with sterile alcohol before accessing the port. To begin, draw 10 ml of citrate anticoagulant into the 60 ml syringe. Then draw 50 ml of blood from the patient, filling the syringe to 60 ml. Inject the anticoagulated blood into the top port of the device. Place the device and counterbalance into the machine buckets at opposite ends. Close the lid and set the machine at 3800 RPMs and 1.5 minutes. When completed, the blood will be separated into a platelet plasma suspension and packed red blood cells. Aspirate the PPS into a sterile syringe. And transfer to the concentrating accessory. Counterbalance the accessory and place into the machine. Process at 3800 RPMs for 5 minutes. When completed, the platelet buffy coat will be separated on the bottom of the accessory device. The rest of the solution is now PPP. Aspirate the PPP, leaving 7 to 10 ml in the device. Attach the 12 ml syringe and resuspend the platelets into the PPP. Tilt the device and aspirate the PurePRP. This concludes our instructional video. For more information, please call 239-481-7725 or send an email to support@insight.com.